Hello, everybody. Welcome to Medical Myths, the show about what not to do with your body. Although today we're not really talking about what not to do with your body, but I guess that the tagline still stays. I was going to do this episode last week. Some health things happened. I'm fine. It was just a migraine and was kind of sitting in my house with all the lights off and was like, if I can't turn the lights on, then it's going to be a problem doing a stream in front of a computer screen with all my lights on. So I just decided to postpone it to today. And I feel like today I'm going to be able to communicate better about this. And as you all can tell, the title of this stream is Are All Babies Female? And this is a debate that I've had to weigh in on more times than I can even count. Because the myths surrounding this are pervasive, and I think the reason why this myth that everybody at some point in their lives have been female is so pervasive. I think in part, number one, because of outdated science, and people have latched onto studies from the early 2000s and earlier, which somebody that I was kind of debating with a bit on Twitter. And then I don't know if it's the same person in my comment section talking about nipple development and why having nipples makes you female. But this person in particular was citing studies, a particular study from 2001, which is a long time ago. We've learned a lot since then. And I think people that have not updated their information are going to perpetuate this myth. And then also, it's just a gross oversimplification of what actually happens in reproductive development. And I think it's very easy for any lay people to see what they learned in a high school biology class or a college biology class and say, well, this is all there is. But you start to dig more into the processes that cause each of these structures to develop, and it becomes way less simple. And this is something that I've really been saying, especially this week, is that the second somebody says that there's no nuance to this conversation, I'm leaving and I'm not interested in talking to them anymore, because anything simple from the surface, and this especially is an issue that is definitely not simple. And I've made videos on this in the past that were a lot more simple even than what we're going to go into today. So if you want to catch up on any of those where I break it down into a bit easier pieces to digest, they're from a long time ago when I first started my channel, when I break down each individual step of the pathway. You can go watch those. And I do have a short video about why female isn't necessarily the default. But today we're going to get into even more depth than we talked about previously. So the claim specifically that I'm debunking is the claim that all babies start off phenotypically female and then develop male genitalia later on. This is not true. This is a gross oversimplification. And I don't even like the term default. And we'll get into why. But first of all, I'm going to clarify what is phenotype. We know that genotype refers to the genetics, the chromosomes, the DNA on these chromosomes that encode, which I don't really like the word encode, but it's an easy enough term to use. I'm using it colloquially. Encodes for the cascade down the line. And so the DNA isn't always going to be expressed, which is why we have phenotype, which is the term defining the expressed characteristics. What we see in an individual is their phenotype. And so I want to clarify that when we're talking about phenotype, yes, we are talking about the external and internal appearance of the anatomy. But I think there's a misunderstanding as well, especially from men, because we're taught most of the time in biology classes, we're taught that like male anatomy matters and female anatomy doesn't matter. And so unfortunately, we have a lot of people entering society that do not understand what goes on with female anatomy. So we're going to clarify that a little bit today. There are distinct structures, and I kind of hate that I have to go into this, but there are distinct structures that we classify as male versus female, and that you can have overlap between these. These are not all of these are mutually exclusive, and especially there is some overlap even among things that are homologs. And we'll get into which structures are homologous. But when I say homologous, it's a structure that starts off indifferent, and it can be one of two depending on the hormonal input. So, for example, the penis is a homolog of the clitoris. They are the same structure, just differentiated a little bit differently. And I think when people think of female phenotype, they think that female is just a vagina, which is just the hole. They think that female is a hole and that's it. And maybe a uterus and maybe some ovaries. But like these are actual structures that have to develop. It is not just a female, it's not just a blank slate. These are structures that had to have had expression throughout the development, and they go through an actual active process to become female phenotypes. So these structures that would altogether give somebody a female phenotype are ovaries, fallopian tubes, the uterus, the cervix, which is part of the uterus, the vagina, and those are the kind of internal structures. You also have external structures such as the labia, the clitoris, etc. So these are all structures, and you can have some of these, none of these. Some of the male structures, such as the penis, the scrotum, testes, vas deferens, epididymis, seminal vesicles, prostate, all of that. And you can have a mix. We're going to focus kind of on people that have all of one set for this particular myth that we're debunking today. But here you go. There are distinct structures between the two. And so now we'll kind of get into what causes somebody to become one or the other. And in my previous videos, I've talked about a pathway that looks like this. And I'm going to pull it up. I've talked about a pathway that looks like this. And this is a still very gross oversimplification. And we're going to expand on this even more. So to simplify this right off the bat, as a fetus, around 6 weeks, you're going to start reproductive development in utero. And everybody starts with indifferent gonads. So you're going to be XX or XY or any combination of X's and Y's from conception. So your genotype is already determined early on in fetal development, but then around 6 weeks, we start to see phenotypes being expressed. But at that early stage, your gonads start off actually at the level of your kidneys, where your kidneys are now. It starts off at this gonadal ridge, and it's going to form into indifferent gonads. So it's going to have still its XX or XY genotype, but it is indifferent. It has the potential to become either. And this is the term, the bipotential gonads. And throughout the developmental process, it's going to differentiate into a testis or an ovary, or you can even have ovotestes, which is kind of a rare presentation. But generally, you're going to have each gonad is going to develop into either a testis or an ovary. And then depending on how it differentiates that way, it's going to further the cascade into the rest of the sexual development. So as we see here, very simply, if I were to just go with this simplified description, you have an indifferent gonad, and with the expression of TDF, which is testis determining factor, you get testes. And then in people that have testes, they're going to produce testosterone, which stimulates the external anatomy or stimulates the internal anatomy specifically to become male. So it stimulates the mesonephric duct to become the vas deferens, etc. So you're going to have male internal structures. Testosterone is converted via 5-alpha reductase to DHT, which then stimulates external male genitalia, such as the prostate, which, yes, that is external, the penis, the scrotum, etc. So along with that, the testes are also going to produce MIF, which is going to signal to the paramesonephric duct or the Mullerian duct to degenerate, which is going to inhibit development of internal genitalia for the female side. And that's how you get a completely male phenotype. On the flip side, we have the indifferent gonad. Let's say that this is an XX fetus that does not produce TDF. So they do not have SRY as a gene, which we'll get into some more nuance about SRY because that's not even the key factor. If I were to say a key factor of sexual development, it's not even SRY, which is kind of mind-blowing because we often think, well, it's this fetus is SRY positive or SRY negative. And yes, that matters, but we'll get into why that matters and how much that matters. But if you have this indifferent gonad that enters the pathway and it does not express TDF, so no testis determining factor, it's going to develop an ovary. And this is where the myth started, because the disconnecting communication between when a scientist says the default pathway, we do not mean that somebody develops female genitalia and then develops male. So, and this is the thing that I have to say ad nauseum. You see here that this is a fork in the pathway. If the claim that we were all female phenotypically first and then developed male anatomy, if I go back to this other screen, that implies, the statement of saying that implies that we developed this whole tract. We developed a uterus, a vagina, labia, fallopian tubes, ovaries, that we developed all of that and then converted over to male structures. And that is not true. Like we're seeing in the diagram here, even this simplified diagram, I think makes that point for me, that this is a fork. You do not develop one and then develop the other, with the exception of the paramesonephric and mesonephric ducts, which develop simultaneously, and then one degenerates. That's the only thing, but those are still early structures. Those are still kind of indeterminate structures that are then going to develop farther on in the cascade. But when a scientist says that ovaries are the default, what it means to say is that this indifferent gonad, in the absence of TDF, is going to become an ovary, which is still an oversimplification because, as we've learned recently, and what we're going to get into, this is not entirely the case. The pathway is not as simple as no TDF, therefore ovary. There are active processes, active steps, active genes, active regulatory factors that will cause ovarian development. So it's not a default, and I'm going to push back on people that say that this is a default. It's an oversimplification at best for people who don't understand the pathway, and we should stop using that terminology altogether. But for the sake of this simplified chart, before we add on the extra layers of nuance, we have no TDF, you develop ovaries, and then these ovaries do not produce MIF, so then you're not going to develop male structures. You're not going to produce a high enough level of testosterone to develop male genitalia, so you're going to develop female. And like I said, this is simple and not entirely correct because it just assumes that you're going to develop the female side in the absence of testosterone, which we really don't see, because the data that kind of shows that this is false is the fact that, like, if you think about it in this binary, you don't account for gonadal agenesis or any dysgenesis along the pathway, because we have examples of people that don't have either set. We have people with lack of gonads altogether. So in people that have neither of these tracks, it's clear that you might have some testosterone in, or you have no testosterone input or minimal testosterone input, but they still don't develop the female tract. So then how do we get these people that don't develop a female tract if that's the default and if everybody is to develop that? That doesn't really line up. So looking more into this pathway, let's kind of add, we had a question for how is the prostate external? The reason why the prostate itself is located internally, but embryologically develops via the pathway of external genitalia. So the same factor, DHT here, that causes the penis to develop, that causes the scrotum to develop, that's the uh, same hormone component that causes the prostate to develop. So, and it's also, I think it is ectodermal derived, whereas the internal male genitalia, and I think all the internal genitalia are endoderm derived. So that's another differentiation between how we classify the prostate as internal versus external anatomy. It's a factor of the developmental hormonal pathway as well as the tissue that it developed from and which germ layer it came from, which I could probably go into a separate video all about that. But let's add some nuance to this conversation because I think we're ready for some more layers. So if anybody wants to follow along with any of these articles that I'm citing today, they are in the description. So if you want to go back and look at that chart I just showed you, the more simplified one, you can go do that. It goes into, I think that page goes into addressing some gender issues related to that particular pathway, which is why they use the simplified one. But for this is a very long paper from basically a free endocrinology textbook. And you can go in the link in the description. And if you want to read even the entire endocrinology textbook, you can by all means do it. It's free for everybody. But this is just the sexual differentiation chapter, and it's very long, and we're not going to go into every single detail because the amount of detail they address here is astronomical and very incredible to get into. But we'll kind of hit the main highlights that are important for this particular myth. So I'll at least read the abstract and the introduction and maybe the bit on the bipotential gonad, and then we'll kind of free form it from there based on how we best feel like we want to communicate this. So if you have questions, please leave them in the chat. I'm going to try to address them. I might not see them right away because I'm going to be looking at the article. But you can follow along if you'd like to in the link in the description. So they talk about the chromosomal sex of the embryo is established at fertilization. However, 6 weeks elapse in humans before the first signs of sex differentiation are noticed. Sex differentiation involves a series of events whereby the sexually indifferent gonads and genitalia progressively acquire male or female characteristics, believed initially to be governed entirely by the presence or absence of the SRY gene on the Y chromosome. So this is where we get kind of that outdated thought that SRY is the key factor. We're going to debunk that today based on new data. Gonadal determination has been proven to rely on a complex network of genes whose balanced expression levels either activate the testis pathway and simultaneously repress the ovarian pathway or vice versa. So we have kind of this is a one time where I like the use of the word balance because you do have at the same time activation of one pathway and simultaneous repression of the other pathway, or in the cases of intersex individuals, you might have incomplete repression or incomplete activation of either pathway, and you might get some in-betweens or you might get some neither presentations. Subsequently, internal and external genitalia will follow the male pathway in the presence of androgens and anti-Mullerian hormone in significant amounts. I will add that because to clarify, and they will clarify as well, that female fetuses do produce AMH in smaller quantities because although it is anti-Mullerian, and this is a question even I had when I read this, was, hold on, but if the role of anti-Mullerian hormone is to degenerate the Mullerian duct, then how do females produce this and have it be necessary for their development? It's interesting because AMH is responsible for proper development of ova and for proper female reproduction. So you do need AMH. Everybody has AMH unless you are unable to produce it. So the name is a bit of a misnomer. Or the female pathway in their absence. Here we review the sexually undifferentiated stage of embryonic development and the anatomic, histologic, physiologic, and molecular aspects of the fetal sexual differentiation of the gonads, etc. They talk about kind of what they're going to go into. So their introduction, I think they say this pretty well. Genital sex differentiation involves a series of events whereby the sexually indifferent embryo progressively acquires male or female characteristics in the gonads, genital tract, and external genitalia. Sex development consists of several sequential stages. Genetic sex, as determined by the chromosome constitution, drives the primitive gonad to differentiate into a testis or an ovary, and we'll get into that particular pathway. Subsequently, external genitalia will follow the male pathway in the presence of. So this is just the same thing that I mentioned, and kind of goes into the simplified pathway that we looked at. So the bipotential gonad. There is no sexual difference that can be observed in the gonads until the 6th week of embryonic life in humans. And they talk about mice because they surprisingly, it is unethical. Surprisingly, it's unethical to conduct studies to like experiment on embryos in humans. So a lot of the data we have is from mice, and people will say, well, but how do we know that this carries over into humans? We have sufficient evidence that mice are similar enough to humans, at least for this particular pathway, to utilize them as an experimental organism. There is data for certain things that we look into to determine, is this an accurate model organism or not? And that's a separate conversation. Undifferentiated gonads of XX or XY individual are apparently identical and can form either ovaries or testes. So everybody has the potential to produce either. And this is the case even in people that are XX or XY. Everybody can develop either based on the regulatory factors involved, hormone influences, etc., is going to determine what pathway these undergo. This period is therefore called the indifferent or bipotential stage of gonadal development. So the gonadal ridge. So as I kind of touched on a little bit, you start off with the gonads at the urogenital ridge. That becomes the gonadal ridge. And then, so this is a precursor to the urinary and genital systems and of the adrenal gland. So these are all kind of related tissues. They're going to form these indifferent gonads, and throughout this process, both sets of gonads, whether you develop ovaries or testes, are going to descend in the abdomen. Testes are going to descend farther as they go into the scrotum, whereas the ovaries are going to stop their descent in the lower abdomen. But they all start around the T10 range. If you think of your spinal levels, the T10 vertebra is where that starts, which is why, interesting fact, like the nervous system intervention, an innervation of the genital tract, and especially of the gonads, is from T10, which is also the part that innervates your belly button and the skin around your abdomen. So that's kind of why, is a bit off topic, if you touch your belly button, you might feel like you're engaging in dirty activity because it's the same nervous system innervation as your gonads. So you feel like your gonads are actually being stimulated when they really aren't. But that's just a fun little fact. So you have that happening, and they looked at birds as well, and the whole Sonic the Hedgehog gene signaling mechanism, which is a really fun gene to look at, that establishes what structures become on the dorsal side or the back side, and which ones become ventral side or the front side of a human. And you have other genes that associate laterality, forward left versus right side, etc. This whole process is very extensive. Um, but here, if you look at this, it goes into the age from conception and what structures develop along this line. So at about 22 days, you start getting the primordial germ cells. And then at 24 days, you get the Wolffian ducts. And when I say the Wolffian ducts are the same as the mesonephric ducts, these are the same structures. We're starting to move away from naming structures after people. So if I say Wolffian duct or if I say mesonephric duct, these are the same structures. And the paramesonephric duct and the Mullerian duct are also the same structure. So don't be confused by that. So then we have these primordial germ cells. These are the stem cells of the reproductive tract. They're going to start in the yolk sac. They're going to migrate to the hindgut. They're going to keep moving to the urogenital ridge, and then they're going to form these gonadal primordia at 32 days for the indifferent gonads. You're going to start developing the Wolffian ducts. You're going to start also similarly right after that developing the Mullerian ducts. And at this point, the urogenital sinus, which is starting to form the external anatomy, and the genital tubercle are going to start forming. And these are terms that I'm throwing out here, which we haven't really gone into yet. So you can come back to this timeline later on if you want to see specifically when these structures form. But I want to pull up what these structures look like just so it makes a bit more sense. So this is what I'm talking about with the bipotential stage. So you have these gonads developing in the fetus. You have the Mullerian duct, which is going to be on the outside, which is why we call it the paramesonephric. Because para meaning next to, and then if the blue here is the mesonephric duct, then the paramesonephric duct is next to the mesonephric duct. So that's kind of also why it's easier to call it by that terminology. But you have both of these tracts developing. And so they are not sex specific at this point. They're still indifferent because both types of embryos are going to have both tracts. So you're not only developing a female internal tract initially, you're also developing a male tract initially, and then one of these is going to degenerate as we go. And there's an experiment that was done here that you can see by these other pictures, which kind of shows the influence of the gonad, especially on the rest of this pathway. Because we know that the ovaries are going to influence maintenance of the paramesonephric duct or the Mullerian duct, and the testes are going to promote maintenance of the Wolffian duct or the mesonephric duct. And they learned this by having a male or a female, and that like an indifferent fetus, and then they essentially chemically castrate the embryo, and in doing so, they caused they still didn't have gonads. So in somebody who so they found a way to, I guess, not produce these gonads in this person, or maybe I'm kind of a bit unclear on what they did, how they did this with the gonad, but either way, or maybe they just removed the testes. I don't know. Either way, they castrated this mouse, and then they learned that in removing the testes or inhibiting the effect of the testes, that you're going to develop these paramesonephric ducts, and you're going to degenerate the mesonephric ducts, which kind of shows that the gonads do influence this pathway. They did kind of similar thing where they had a female organism and they grafted a testis onto the female, and they got maintenance of the mesonephric duct on the one side that the testis was on, even though they still maintained the ovary on that side. So it's a little bit more in depth there, but that's kind of how we learned that this even happens. But this is what the internal tract looks like initially. And then I'll briefly show you the external side before we get into the DNA, because as you can see from scrolling through this is a lot of DNA to cover. And I promise I'm not going to go through all of it today. We're just, here, maybe I can just go to, here we go. So this is the other component to demonstrate the indifferent nature of the fetus. So we had the indifferent gonad, which is the beginnings of the internal tract, which then, yes, will cascade to develop the external tract. But you have indifferent external genitalia as well. And this is what we call the urogenital sinus. So the urogenital ridge is the internal component that's around the kidneys. The urogenital sinus is going to start at the perineum and is going to develop. So if you have these two sides of the reproductive tract, you've got the internal side, the external side, and they're going to kind of meet in the middle to make one cohesive tract. So the urogenital sinus is going to start looking very ambiguous. It's very in between, and this starts forming around 9 weeks. So you can see the urinary bladder here. You see the urogenital sinus developing, which is just the opening that's going to become the urethra in the male. It's also going to become some of this is going to relate to ducts and associated with the prostate and other glands. And then is also going to develop and differentiate into the vagina in females, which is then going to close off and separate the urethra from the vagina in those females. So, but you can see that all of these structures are present, but you don't have things like you're missing a prostate at this point, you're missing a uterus at this point. You can see here the Mullerian duct kind of showing. You can't see the Wolffian duct here, but this is very indifferent. And from the external side as well, you can see the genital tubercle, which I kind of want to use a different picture for this because I have on the other page, I have the same diagram, I think, that shows these as homologs. Yeah, and I'm gonna clarify because yeah, Matt Walsh weaponizes this phrase, but that's not it at all. Matt Walsh will use this phrase to talk about puberty blockers. I'm not using it to talk about puberty blockers. I'm using this to say an actual like genetic shutdown of the process, which is not what's happening when Matt Walsh is talking about it. He's misusing the term. But we'll look at the specific homologs of sexual anatomy. So we have, and I know that this episode's probably going to be very easily demonetized because of what we're talking about in the images that I'm showing. But this one color codes it kind of the same structures to show what structures become what on either side. So we have this undifferentiated stage, and we have the genital tubercle, which is this whole area, including some of the purple and some of the pink, which you can see the purple is the glans area, which is going to develop into either the clitoris or the glans penis, depending on which pathway. You have the urogenital fold, which is going to fuse together in the male tract at about 12 weeks to become the shaft of the penis, and it's going to stay open and not fuse together for the female side. And you're not going to be able to see that. This is kind of the more internal aspect of the vaginal opening. And you're going to see that the labia are going to cover this. And that's why you see the green predominating in that picture, because this would be a scrotum if we went to the male side. But these are clearly distinct structures. And at this indifferent stage, this is not female. Like, you can look at this, and this debunks the point that we were all female first, because you don't develop this female tract and then go to the male. You have this undifferentiated in-between stage that becomes one or the other in this fork of a pathway. So I hope that clears that up a little bit. Yeah, just say no to gonads. Just say no. So let's get into the nuances of sexual differentiation and why I, in this article, does not accept necessarily that SRY is the most important factor in sex determination and testicular function. So here we go. So in I'm not going to read this verbatim because they go into a lot more detail than I think I'm going to. But in the simplified diagram here, let's go back to that because I'm going to recap it really quick just in case uh, I need to reiterate. So in this simple pathway, they have. Oh, and here's a simplified diagram as well. So here's the simplified explanation that many people have heard about SRY is that males possess the SRY gene, which is located on the Y chromosome, and then SRY encodes for testis determining factor or TDF, which once you produce a testis, that you're going to produce MIF or Mullerian inhibiting factor, which causes degeneration of the paramesonephric duct and the inhibition of the female pathway. Testis also produces testosterone, which promotes development of male sex characteristics, as we saw in that pathway of differentiation previously. And then they're making the claim as well, again, very simplified, that females do not possess SRY since they don't have a Y chromosome, they don't have SRY, and so are not going to produce TDF. So this, again, like the simplified form is going to say, if you have XY, then you're going to have most likely the SRY gene. And then if you have the SRY gene, you're going to produce TDF, and then the rest of the pathway is going to follow, which sounds great. It's a good entry level way to think about it, because adding these other layers complicates things. But I think it's worth going into to highlight how nuanced the process is, because SRY is not the only factor that matters. And I might not even read it because it's like look at this chart and you see like all of the known genes, and this is just the known ones as of 2020, the known genes that influence this pathway of development for gonadal development. So this is just for developing a testis or an ovary. This is not even considering the external anatomy or anything else. This is just the involvement in developing a gonad, which is pretty intense. And there are a few different diagrams that we're going to use, and I don't know if I want to use that one or if I don't want to use this one. This puts it all together. We might come back to that one. Okay, so we'll start with this diagram, which looks a lot more intense, right? So we had that simplified thought process, and now we're adding. You can see we're adding a lot more steps to this. So we have this undifferentiated gonadal ridge. We have this indifferent gonad, and SRY, as we see here, we're going to focus right now on the XY side, and we'll get into the XX side momentarily. But we see on the XY side that SRY is present. And yes, so this is important, but more important than that is SOX9. And I'm going to argue, and this article entirely is also going to argue, and that's the reason why I'm arguing it, is because I'm convinced by their explanation here, is that SOX9 is the primary regulator of this XY pathway and is going to determine a testis phenotype in a fetus. So SRY, what's going to happen is in the XY fetus, you're going to have expression of the SRY gene, which is going to be a transcription factor for SOX9. And so you're going to get initially, SRY is going to be produced and repeatedly stimulate SOX9 production, because at early stages, SOX9 cannot produce itself. But we're going to end up getting to a cycle with SOX9, PGD2, which is prostaglandin D2, and FGF9. So SRY is going to stimulate the initial production and upregulation of SOX9. So SRY is expressed, and you get this production of SOX9. You're ramping up production of this SOX9. And then there comes a point in the development, and I'm not sure, they specifically say in here which week SOX9 starts expressing itself. And I don't remember off the top of my head. But you have this stimulation of SOX9, and then eventually you get to a point where you have enough SOX9, because SOX9 is, number one, going to stimulate its own production. So SOX9 causes more upregulation of SOX9, and you have SOX9 stimulating prostaglandin D2 and FGF9, which then stimulates more production of SOX9. And then this SOX9 is going to inhibit DAX1, FOXL2, WNT4, beta-catenin, etc., and a bunch of these pathways that you see on the XX side, which we'll get into in a more detail in a little bit. So SOX9, not only does it have the important characteristics and regulation of these other genes and transcription factors that cause the stimulation of the coelomic vessel, causing testes. So you have, number one, the ability to produce testes from SOX9, and you have repression of the alternate pathway. And so that's why SOX9 is going to be so essential for development of testes. And so we end up seeing this, how we learned that SOX9 was more important than SRY was that we have examples of people, and this is actually an example I used on transatlantic. And when I brought up the example on transatlantic, I don't know if SOX9 was specifically the factor that was different in this particular clinical case. I don't know if the researchers on that clinical case know what the particular variation was in that case, but we have case reports of people who are XY females that have SRY present. So it was thought that if you had an SRY translocation, then that was the only way to be an XY female, someone with XY genotype and a female phenotype. Yeah, and the plot twist, SRY. So we had a case. Well, I didn't have this case, but these people had a case where they had a teenage girl who was being brought in for amenorrhea, so didn't have a menstrual cycle, and she was in her kind of late teens. And so mom brought her into the OBGYN and said, "Hey, what's going on with my child?" And they karyotyped her, found out that the daughter was XY and was phenotypically female. And that is why the daughter did not have menstruation. But they karyotyped the mother as well. And they found out that the mother was also XY female, which is wild, because initially looking at this case, number one, we would expect that this person that was XY would not have an SRY gene in order to be a female phenotype, but the mother did have SRY. So some other factor was involved to cause the female pathway to develop despite the presence of SRY. Also, this XY female had, I don't know if this was the one that had 2 unassisted pregnancies, but I know this person at least had 1 unassisted pregnancy. There may be another case that had 2, or maybe this is the same one. But had an unassisted pregnancy. So they only found out about the XY karyotype from after having the daughter complain of amenorrhea. So the mother had no idea that she was XY until she got karyotyped later, like after her daughter was a teenager. And there's some chimerism as well with that. Kind of an interesting situation, but SRY was present. So in these cases where we see XY females that have SRY, what is the mechanism that would cause somebody to still become a female phenotype in the absence in the presence of SRY? And so I think, not knowing what the outcome was of that case, I think it was related to SOX9 upregulation, because we do see in other cases as well. Godless Granny asked an important question, and this is a super chat. Thank you for the super chat, Granny. An unassisted pregnancy is they had no in vitro fertilization, no need for a sperm donor or anything. Like, this was like the person had sexual intercourse and they organically had their own baby without other medical intervention. So it's pretty wild. Like, oh, hi Spooky, what's up? Yeah, so it's a pretty wild case. So we have seen in that case most likely, and in other cases, that, and does this mean hypothetically her eggs could have only Y chromosomes? It's potential. I don't know if they did an ovum analysis on her. It's possible. I don't know the extent of her chimerism, but it's definitely something that I'm curious about. But we do see, kind of going back to this, with people that have had an absence of SRY, some of them have still developed testes. Like, you can have people that are XX and have a gain of function mutation in SOX9 and then develop along the male pathway and have testes. So that suggests that with enough upregulation of SOX9 and with a gain of function mutation of SOX9, that is going to primarily determine the gonadal outcome for the fetus. So that was a lot, talking about the male side. And then talking about the female side. So getting into that with SOX9. So looking at the XX gonadal ridge, we have somebody that does not have SRY. And like I mentioned, SRY is kind of only necessary to stimulate SOX9 production. But you kind of need, at least in most cases, people do not have enough SOX9 to cause that auto-stimulatory cascade. And so that's why we see, in the absence of SRY, the majority of the time, you're not going to get SOX9 upregulation. But that's not the only factor. Because, and this leads into the data that supports that fetuses are not female first, and that female is not the default, is that they saw with fetuses that were XX, without other genetic factors, even if you were, if you didn't have SRY, which we're going to assume you don't in this XX model, even in the absence of SOX9, if you have the absence of a bunch of other factors, namely WNT4, FOXL2, and RESPO1, which RESPO1, I guess, is a stimulatory factor for WNT4, but probably if we're looking at the XX pathway, WNT4 is kind of the key factor equivalent to the SOX9 of the XY pathway. So WNT4 is shown to be necessary in certain concentrations to stimulate the female pathway. So if you have XX and you don't have SRY and you don't have SOX9 upregulation and you don't have WNT4 upregulation, you're not going to develop a female tract. You're not going to develop ovaries. You're going to develop kind of nothing. You're going to have dysgenesis or agenesis of the pathway, and you'll have an indifferent baby, an indifferent person in general. So the default, if we're going to really assign a default, Female is not the default. The default is neither. The default is dysgenesis or agenesis. So it's not a binary in that you are either a testis or an ovary pathway. You could be both or you could be neither. And having the absence of one does not mean that you are the other. It could be that you are neither. And I hope that makes sense. It kind of goes against a lot of the logic that we're taught. But I was trying to see if anybody had any questions before moving on to more of the XX pathway. But we're kind of, is it true that women have stripes? What is that about? That is an interesting question. So let's get into the XX pathway, because I do also, we're coming up on the hour, and I do want to talk about breast development because that was a key thing in the debate on the internet. So let's look at the XX pathways. So we have, again, an XX fetus. We have no SRY, so we don't have the proper cascade to upregulate SOX9. And we know that SOX9 cannot replicate itself initially. It's going to take some time to ramp up SOX9 in order to do that. So we don't have that stimulatory factor, and we have inhibition of SOX9. So the factors that are going to do this are RESPO1 is a transcription factor, which is going to encode for WNT4 and beta-catenin. And WNT4 is going to actively inhibit SOX9. FOXL2 is also directly going to inhibit SOX9. You can see here DAX1 has an inhibitory factor on SF1, which is, we're not going to go into that pathway. And then you have FST, which is going to be inhibited, or which is going to be stimulated by WNT4 and beta-catenin, and stimulate the activins necessary to develop ovaries. And something that's interesting about WNT4, which does need to be mentioned, is that WNT4 is not only involved in ovarian development, it's also involved in intestinal development and adrenal development, and I think kidney development and lung development as well. So in people that are missing WNT4 in significant amounts, or have some variation in their WNT4 pathway, whether it be the enzyme itself or the receptors for WNT4, you not only see ovarian dysgenesis, but you also see adrenal dysgenesis, renal issues. So you see kidney dysgenesis or agenesis, and you see some lung issues as well, potentially also some intestinal issues. So you might see broader issues coming to play when you talk about an absence of WNT4. So it's a bit more complicated than just WNT4 ovaries. That's it. There's whole separate pathways with that. And then RESPO1 is not entirely 100% necessary for WNT4 activity, but like in kind of in the way that SRY upregulated SOX9, RESPO1 is going to be an upregulatory factor for WNT4. So that's how you're going to get enough production of WNT4 to suppress SOX9 production and stimulate ovarian synthesis. So, yeah, Alma, I love that this is the question. Yeah, I'm going to say that women do not have to have stripes. Some women might have stripes and some women might not have stripes. And that's all I'm going to say. All women are great. If you have stripes or not, you are amazing. And so if you want to read more about this pathway, this article goes into a lot of detail on all of these genetic factors And then talking about more specifically the beta catenin pathway, how this how WNT and RESPO interact with each other, and how you get, because this is a G coupled protein receptor, which is a mechanism common to a lot of cell signaling mechanisms. It's how a lot of medications work by G coupled protein receptors. It's a fun thing to look into if you're interested in cell signaling. But if you want to look more at how WNT and RESPO do this, you can look at that graphic. But here's the kind of one that I wanted to look at as well, because this puts all of this together. So we'll go, because I think repetition might be helpful to, because this is also going to show Sertoli and Leydig cells, which contribute to the specific hormonal inputs involved in specific structures. So we have the mesonephros, which is the primordial tissue that is going to end up becoming the gonadal ridge. You're going to have the coelomic epithelium, which is kind of our stem cells that are going to become the indifferentiated gonad by means of all of these genes and transcription factors on in the black here. Those are all going to create this undifferentiated gonad, and then we are going to see whether or not there is an SRY. If there is an SRY, for this first example, we're going to follow the pathway with SRY. SRY is going to code for TDF. All that's going to stimulate the production of SOX9. SOX9 is going to promote development of the testis through production of itself and through this upregulation of FGF9, PGD2, and this little cycle here stimulates a testis. And through that, you're going to have differentiation of two different cell types. You're going to have the Leydig cells, which are going to produce testosterone. And testosterone is going to tell the mesonephric ducts or the Wolffian ducts that we're going to keep these. They're going to develop and become, with testosterone influence, the epididymis, the vas deferens, and the seminal vesicles. And so that's kind of the internal tract, including the testis, and the differentiations of the Wolffian duct are the internal tract. And then testosterone is going to be converted by 5-alpha reductase to DHT, which is a more potent type of testosterone. And that's going to act on the urogenital sinus, which was that indifferent external genitalia. And it's going to tell that to go down the male pathway. You're going to develop a prostate, and the penis, the scrotum, the all of the external anatomy, etc. So that's kind of putting that all into a summary for the testis side. Let's go back over to the ovarian side. So once again, we have the mesonephros becoming the coelomic epithelium, becoming the undifferentiated gonad, and this time we don't have SRY. So in the absence of SRY, we don't have enough SOX9 to regulate its own production. So in that case, we're going to produce, in this person that's going to have RESPO1, is going to, and I didn't mention the Sertoli cells and the AMH and inhibition of Mullerian ducts, but you can read that pretty simply on your own. So we have the upregulation of WNT4 via RESPO1, and this interplay with beta-catenin and FOXL2. All of that stuff is going to suppress production of SOX9. And in the absence of SOX9, we're going to develop an ovary. We're going to produce follicles, which are going to produce estrogen and progesterone. We're going to see in the absence of testosterone, we're going to see regression of the mesonephric duct and persistence of the Mullerian duct via estrogen stimulation. We have fallopian tubes, uterus, and the upper vaginal canal. And then this undifferentiated external genitalia are going to differentiate into the female side of the pathway. So that's a summary, kind of everything that's really important about this. But I do, with the last kind of half hour, we're going to, oh, bye, Granny. Good to see you. We're going to look into breast tissue because this was the other highly, uh, I could say contested, but it's really not because the scientific consensus is very clear. It's people that don't understand the consensus and haven't read past a grade school level of science that are going to say that nipples mean that we were all female. So let me quickly explain the argument that was given to me, and this is really funny. So I get somebody on Twitter, of course, and then I think it's the same person, might be a different person, because I do actually see this discussion a lot in the comments section, saying that, well, I believe that all fetuses started off as female because why would males have nipples if that weren't the case? So let's look into this claim real quick because I disagree with that because we just looked at the entire pathway for sexual differentiation in the fetus. And breast development is a completely separate pathway. It is not at all involved in the sexual differentiation pathway. So even if you're going to make the simplified argument that female is the default pathway due to the lack of SRY, it still does not explain or account for nipples in males. And so you might be asking, why do males have nipples? Why do male humans retain this? And I was actually talking to a friend of mine who is a veterinarian, and she kind of looked at me funny and was like, yeah, this is an obvious answer that humans are mammals. Like, this is a trait of mammals. Like, this is a defining characteristic, in fact, of mammals. And she's gotten patients in her clinic who were male cats, and the owner's like, "Hey, I've got my cat, and he's got like this weird mass on its abdomen. Can you tell me what it is?" And she'll tell them, "Well, this is a nipple." And they're like, "But it's a male cat. How can my male cat have nipples?" Well, your male cat is a mammal, and the majority of mammals have nipples, and a lot of male mammals have nipples. A lot of them you just can't really see. Um, but especially in primates, you see male primates with nipples, and it's because of this separate pathway. Nipples are they are sex neutral characteristics, and the person in my comment section laughed at me when I said, like, because my response to them, to their question of, then why do males have nipples? That's obviously a female trait. And I said, no, the reason is because nipples are a sex neutral trait. And he laughed at me and said, like, LOL, okay, as if that was evidence of the claim that nipples are a female trait. Um, but they're not. Like, males and females, I'm going to say the majority because, of course, there are always exceptions to everything. But the majority of males and females all develop nipples, and you can still be male with nipples. But let's look at this next article. This is from Seminars in Plastic Surgery, and this was from 2013, so it's a bit older. But our knowledge on breast tissue, like, this process is way simpler than sexual differentiation in utero. So we know a lot more about this than we still even know about gonadal differentiation. But yeah, I mean, even in their abstract, they talk about mammalia are so named because based on the presence of the mammary gland in the breast. And they talk about the mammary gland as an epidermal appendage derived from apocrine glands. And it's got parenchyma and stroma originating from ectodermal and mesodermal elements. So these germ layers in fetal development, we have the parenchyma of the breast is going to come from ectoderm, the outermost germ layer of the fetus, and then the stroma is going to, so the stroma or the Glandular tissue is going to come from the mesoderm or the mesoderm, however you want to pronounce it, which is the middle layer, hence the meso components, respectively. So you're going to have mammary glands that produce milk. And an argument that is used is that, well, males don't lactate, so therefore this is a useless structure. And that's not entirely true because so being human is sex neutral. Absolutely. I agree with this statement. Being human is sex neutral. Thanks for the memories. Oh no, we got a comment about G-Man. G-Man thought nipples were magnetic and aided in detecting direction. Wow. That's a take for sure. But for the example of talking about lactation, it's actually not true that males can't lactate. And the reason is that, and they'll get into this here. I guess we'll get into that a bit later. But breast tissue forms. Actually, I do want to read this because the way that they say it is great. This is not what I wanted. There's a particular statement. Here we go. Okay. Sexually dimorphic development of the breast first begins at puberty, and unlike the preceding stages of development, pubertal changes are heavily under the influence of sex hormones, in particular estrogen. So the reason why we have nipples and the ability to lactate as males is because this is a change that happens at puberty, and it is under the influence of estrogen and prolactin. And we do have examples, if you've heard of cases of galactorrhea. This is where males lactate. And this often comes from a combination of high estrogen levels and high prolactin levels. Or even if, like a lot of times, this may come from a pituitary tumor, like a prolactinoma. So if you have a pituitary tumor that produces prolactin, then you can get males that lactate. And then you have to hormonally suppress that pathway and preferably, hopefully, you remove the tumor. But a lot of times you just watch prolactinomas because operating on the pituitary is kind of a sketchy thing to do because you could mess with a lot of processes. So males can lactate because the ducts and the functionality of the breast is sex hormone stimulated. And we do have examples of trans women who went through a male puberty that are able to lactate with the appropriate amounts of HRT. And granted, because of this, because of going through a male puberty, they don't develop the same extent of gland structure that that someone that went through an estrogen dominant puberty would have. They likely are going to produce less milk than a cis woman, but you can still get production, and it still often offers a lot of gender euphoria and quality of life improvement for mothers who are trans. So that's kind of an application of this that demonstrates that this is possible for people of that background to lactate, and also that we can use that to improve quality of life for trans people. But yeah, a lot of this happens in puberty, and I think we'll go back a little bit and look at the development of the breast tissue in utero. So they talk, I might just read this to y'all. So Yeah, here we go. Here's another, and they use the term gender kind of outdatedly, um, which I'm going to ignore that because the rest of their data is appropriate. They just kind of are not, and this is also not an American written article, so there might be an English barrier as well. So of note, prenatal human breast development does not differ between the sexes. So they are not different. They are the same in the prenatal development stages. So as early as 4 to 6 weeks of gestation, mammary-specific progenitor cells may be seen. Around day 35 of gestation, proliferation of paired areas of epithelial cells in the epidermis of the thoracic region occurs. These discrete areas of proliferation extend in a line between the fetal axilla and inguinal region and form two ridges called the mammary crest or the milk lines. So you develop in utero, you kind of develop this initial milk line. You develop initial mammary glands. The initial functional structures happen in utero, but they don't fully develop until puberty. And we even see they mention in this article too that uh, the mammary glands develop in someone in an estrogen based puberty or and somebody with the ability to menstruate, you get additional mammary glands forming with every subsequent menstrual cycle. So you're getting extra glands additions from menstruation, and you're going to get extra glands from pregnancy, from progesterone and the prolactin production involved in pregnancy, which is why you're going to get significantly more milk production, uh, like after pregnancy, because uh, that's kind of how the pathway works, and what ends up being ideal for the fetus because you want the fetus to be able to feed right after pregnancy. That's kind of the whole point of mammary glands at all. So that's kind of why, as well, you'll have the initial gland present in utero that both sexes will uh, have, and then why you're going to get differences in production after puberty because of this continual addition of glands throughout puberty for females and not that way for males. But the development uh, further differentiates like in throughout pregnancy all the way through third trimester. And this is an interesting point too that yeah, here we go. The infant breast. So um, this is something interesting too that I'll mention with uh, all fetuses, but then leads into something as well. It's kind of interesting for female fetuses. So once you're born, you've gone through this whole differentiation process of the nipple and the breast tissue, and then you have from the maternal parent, you're going to have high amounts of estrogen in utero, and then once you're born, that level is going to continually fall. So you're going to have uh, a baby that's used to high levels of estrogen, and so you're gonna get some biological function in the fetus that is reflective of this high estrogen level. And what we see here is that we see in as many as 70% of term neonates, you see unilateral or bilateral breast enlargement and or transient secretion of milk. So you can have babies that lactate uh, in their neonatal stage because of this high level of maternal estrogens. And this is in not sex specifically female neonates. This is in 70% of all term neonates. And they mentioned earlier that you really don't see this in such high numbers in preterm infants, which has some implications maybe for the process and when this estrogen level peaks in pregnancy and all that. But you have milk production in male babies that are just born. Uh, and then this breast tissue is going to kind of degenerate and go back to this kind of indifferent breast stage for the babies. And then this is also interesting because you get for female babies, the fun point that I wanted to bring up for female babies is that you can have female babies that menstruate as neonates because of this high estrogen level in utero. You expel them from the uterus, so to speak, and before this estrogen has time to leave their system, they're going to have a menstrual, not really cycle, but they're going to menstruate. So if you have a very new neonate that's female and that is bleeding from their reproductive organs, that is actually probably menstruation and probably not problematic. You can still and probably should get them evaluated, and if that's happening, your pediatrician will probably know about that because they should be evaluating for, I mean, anything problematic that's going on in your baby. But that is normal. It is normal for female babies to menstruate, which is kind of a mind blowing thing. So I think I don't know how much more there really is to say about uh, the specific claim about breast tissue. But here, if you wanted to see the functional changes in breast tissue throughout development, you can find that here in this article as well. You can read more about the changes that happen during puberty for the female breast or during female puberty, and yeah, they talk about the Tanner stages and different cellular changes that occur. It's a fun and fascinating uh, article to read. They also talk about pubertal male development, and they say, and this is something of note as well. During puberty, up to 40% of those assigned male may develop transient gynecomastia, uh, which is possibly due to higher levels of estrogen. But in that case, yeah, you see it's not necessarily lobular, um, but you do see this. You do see gynecomastia, and with enough prolactin, you could see them lactate. So that's pretty cool. And they go into specific regulation of breast development, like how these hormonal factors play into it and all that. It's really cool. So you can, in the description below, you can find all of the articles that we looked at today. You can find that giant uh, endocrinology online textbook for free if you want to go torture yourself for a little bit and read that. I've got the more simplified explanation, which I think it's okay. I think it's okay uh, to use the simplified explanation and the simplified model as long as we understand that it's not entirely correct. Because I don't think that everybody needs to be an expert in embryology. I don't think that that information is useful. And especially, I don't think people like, I think muddying the waters with too much nuance is difficult and can maybe cause more misinformation. So I think it's okay to use simplified models, but I think it's important that if we're going to use a simplified model, that we communicate that it is simplified and that this is not the full extent of everything that's out there. And I encourage teachers, especially science teachers, in high school, even in college, uh, that like especially at these earlier educational levels, I'm not gonna take that intense genetic outline and show a ninth grader they're not going to understand it. It's going to be confusing and they're not going to learn. So education follows a pathway of, we're going to give you this simplified explanation so that you understand the concept of what we're talking about. And then in the next level up, I'm going to, clarify a little bit and say, hey, this was simplified. Let's add some more layers. Like, let's add a little bit more nuance and complexity. Like, okay, cool. That's awesome. And it's still simplified. And then you add maybe a master's degree level of information. Like, okay, this is a lot more nuanced and a lot less simplified. And wow, this is really complicated. And then you get into a doctorate level and into the new cutting edge of the research, and you say, hold on, like this is not even close to as simple as I was taught. And it's mind blowing. It's really cool to learn this, which is why, like, when somebody shows up to these debates and says that I believe that all babies are female because of this ninth grade level of simplification, like, well, no, you're relying only on basic biology, and you have to move past that. At some point, you have to move past a basic level and adopt a more advanced way of thinking. And so I think it's okay to say, like, this is the simplified process, but that is not all there is, and use that to spark curiosity. Like, maybe give a little taste of the next level. Like, teach them, like, yeah, okay, there's XX and XY, but did you know that there are different genes on these XX and XY that can be different in different people and cause different outcomes? Like, no, I didn't know that. Let's learn more about that. And then you can add some more nuance on that. Like, yeah, and so this gene is called SRY. And if you see SRY changing from the Y chromosome to the X chromosome, then you're probably going to see uh, sex reversal in the fetus. Like, oh, wow, that's super cool. And then that's the next level of nuance. And then you add on what we talked about today with, okay, yeah, so we talked about SRY, and for the most part, SRY translocation causes this presentation, but sometimes it doesn't. And there's this other gene called SOX9 that adds this other layer. And also, there are things called WNT4, and you can start adding more things. Like, use maybe a little bit of nuance to get people curious and wanting to learn more. So there is an appropriate time to use the simplified explanation, like we did earlier, as long as we understand that that's not the whole picture. So I know that was a little bit of a tangent and a rant. Does anybody, if anybody has any questions from all of this? Yeah, and Carrie makes a good point that the most basic level that you see is it takes seed and egg to make us all. We are both products of both equally. Yeah, and that's, I think there's definitely different levels of nuance that you can add, and it depends on what conversation that you're having for what level of nuance you're going to use in that particular conversation. Um, but again, when we're going to, I guess this is a different layer. Like, for people that only utilize the simplified explanation, like, understand that we cannot make decisions, medical decisions, political decisions, etc., based on this simplified definition. And that's why I think that scientists need to be involved in this conversation. And that's why it's not an appeal to authority to say, um, I'm an expert, I've studied this, I have understanding of this. It would be an appeal to authority to say, I'm an expert, so therefore I'm right. That's an appeal to authority, and that's not what I'm saying. But somebody that has studied the nuance and has studied the up-to-date cutting-edge research is going to have a better fundamental understanding of the relationship of these variables. And with somebody that is an expert, number one, I expect them to have that expertise. I expect them to be able to explain to me the whole process. I expect them, if someone says, I have taken embryology classes at the doctorate level and I'm an expert in embryology, I would expect them to be able to explain to me that giant genetic article that we looked at earlier. So if I were talking to somebody, and that's what, like, when I say that I've studied this, I try not to just leave it at I've studied this, therefore I'm right. I try to say, I've studied this. Here's the data that I've read. Here's the process as I know it. Here's my understanding of the nuance. And then I'd ask the other person, like, have you studied this? And it's not that, like, by not having taken a class, that means that by default you're wrong. But it does mean that you probably, if I asked you, can you explain the process of how sexual differentiation happens? And they give me some simplified explanation. It's like, okay, you don't understand it. You don't understand the process. And so I think this is a good takeaway as well, kind of summing up our debrief. If you have somebody making a claim about biology, about trans issues, about anything, if somebody makes a claim and you're not convinced that they have enough information to make that claim, you can, yes, asking for citations is helpful, but also I think something that puts them more on their toes and more understanding of their own pitfalls is to ask them, can you describe this process to me? Can you describe the mechanism of how this happens? Because if somebody is a layperson and they are well read and they have done studying, then that layperson should at least be able to communicate to me what they understand and why that thought process is the way that it is. So I totally endorse entering conversations and saying, can you explain to me how you believe this process works? And then you get a feel for how they think, where they went wrong, and then you can address specifically why they went wrong. Radio Lab, yeah, I haven't watched them yet, but it sounds like they do some good stuff. Yeah, and this is a very in-depth process. So I hope everybody liked this video. Feel free to, again, go into the discussion or the description. I cannot speak. Go into the description and look at some of these articles. If you want to learn more, you can go back and always watch some of my previous videos that break it down more in a stepwise fashion. If that's something you're more interested in, my voice is a bit squeaky back then, so just pardon that. And for a couple announcements, we do have the after show today. So for channel members, and if you are a Twitch subscriber as well, if you're a Twitch subscriber and don't have access to the link to the after show, let me know on Twitter, or even on Discord, and so I can get you that link. But for all of the patrons, channel members, and Twitch subscribers, we are going to hang out in the after show today. There's a separate link, and I hope to see you all there. Something else, we have trivia usually on Fridays at 9:30 over on Twitch, 9:30 p.m. Eastern time on Twitch. And we don't have that this upcoming week because I will be in the UK hanging out with some cool people. So we also will not have Medical Myths next week. So next week is a week off. Enjoy your time doing something else. But go to the after show today. But then after that, when we get back, we're having a very fun event over on Twitch. And I actually need to put this poster up here, but we're having an awards ceremony over on Twitch called the Glizzy Awards. And we are currently taking nominations for certain awards, and it's going to be a whole big thing. I'm very excited. Here's the flyer for the Glizzy Awards on May 26th at the same time that trivia usually is at. So come hang out for some glizzies, for some fun highlight reels. It'll be a great time. So join us over there on that day. And thank you to my lovely supporters on this YouTube channel and in the Patreon. I hope to see you all in the after show today. I don't know what we're doing in the after show, but it's always a great time. So come hang out in the after show. Thanks again, everybody, for being here. And I will see you all in the next one. Bye, everyone.